Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa, where I take difficult biological concepts and make them easy to understand, so you can feel confident mastering the science behind how your body works. Today, we're tackling one of the most fundamental topics in neurobiology, how neurons fire and communicate using action potentials. These tiny electrical signals are behind everything your nervous system does, from controlling your muscles to processing thoughts and memories. Neurons are electrically excitable cells. This means that they can generate and transmit electrical signals. They communicate with one another using two main types of electrical signals: graded potentials, which are used for short-distance communication, and action potentials, which are used for long-distance communication. In previous videos, we explored muscle action potentials, which occur in the muscle fiber itself. But before a muscle can contract, it must first receive a signal from a neuron across the neuromuscular junction. In this video, we're going to focus on nerve action potentials, the electrical signals that occur in the neuron itself and are used to send messages to other neurons, muscle fibers, or target cells. To understand how action potentials work, we first need to understand ion channels and the resting membrane potential. Ion channels are proteins in the neuron's plasma membrane that allow specific ions to move across, following their electrochemical gradient. This gradient combines a chemical difference, where ions move from areas of higher concentration to areas of lower concentration, and an electrical difference, where positively charged ions, or cations, move towards negatively charged areas, and negatively charged ions, or anions, move toward positively charged areas. As these ions move, they generate an electrical current that changes the membrane potential. Ion channels have gates that open or close in response to different signals. There are 4 types of ion channels that are used to produce the electrical signals of neurons and muscle fibers. They are leak channels, ligand-gated channels, mechanically gated channels, and voltage-gated channels. Let's first talk about leak channels. The gates of leak channels randomly alternate between open and closed positions. They are ion-specific. Neurons have many more potassium leak channels than sodium ones, and potassium channels are leakier, making the membrane more permeable to potassium than sodium. Ligand-gated channels. These channels open up and close in response to the binding of a ligand. Ligands can include neurotransmitters, hormones, or specific ions. For example, as we learned in muscle contraction, the neurotransmitter acetylcholine binds to ligand-gated channels on a muscle cell and opens cation channels that allow sodium to diffuse into the cell. Mechanically gated channels. The gates on these channels open or close in response to mechanical stimulation in the form of vibration, touch, pressure, or tissue stretching. Voltage-gated channels. The gates on voltage-gated channels open in response to a change in the membrane potential or voltage. So let's take a look at the neuron at rest. Even when a neuron is not sending signals, it's still electrically charged, much like a battery. This occurs because of an uneven distribution of ions. Sodium concentration is higher outside of the cell, where potassium concentration is higher inside the cell. And thanks to those leak channels, especially potassium leaking out, the inside of the cell stays negative. This separation of charges between the inside and the outside of the cell is a form of potential energy known as the resting membrane potential. In neurons, the resting membrane potential ranges from -40 to -90 millivolts, with about -70 millivolts being the most common. The negative sign means that the inside of the cell is negative in relation to the outside of the cell. A cell that has a membrane potential is said to be polarized. A graded potential is a small, localized change in the membrane potential that depends on the strength of the stimulus. It can make the inside of the membrane more negative, bringing it further from threshold, which is called a hyperpolarizing graded potential. Or it can make the inside of the membrane less negative, bringing it closer to threshold, which is called a depolarizing graded potential. We'll talk more about threshold in a bit. A graded potential occurs when a stimulus causes mechanically gated or ligand-gated channels to open or close. Since these channels are found on the dendrites and cell bodies of a neuron, the dendrites and cell bodies are where graded potentials can occur. The strength of a graded potential depends on how long those ion channels stay open or closed, which determines how many ions cross the membrane. Because they spread out only a short distance before fading, a process called decremental conduction, graded potentials are only useful for short distance signaling. A graded potential can become stronger and last longer by combining with other graded potentials nearby. This is known as summation. If two hyperpolarizing graded potentials summate, this would result in a larger hyperpolarizing graded potential. If two equal but opposite graded potentials summate, one hyperpolarizing and one depolarizing, they would cancel each other out. If a depolarizing graded potential brings the membrane close enough to threshold, it can trigger an action potential. An action potential is a rapid, dramatic reversal of membrane potential that spreads along the neuron's membrane and then returns to the resting state. Let's talk about the steps that occur during an action potential. If you remember, a neuron in its resting state maintains a negative charge inside the cell compared to the outside of the cell. This is its resting membrane potential, and it sits at about minus 70 millivolts. A stimulus generates graded potentials that depolarize the cell membrane, making it less negative. If this depolarization is enough to reach a critical level known as threshold, which is about minus 55 millivolts in many neurons, an action potential is triggered. If threshold is not reached, the signal fades out through decremental conduction. This all or none principle means that an action potential will fire completely if threshold is reached and will not fire at all if it isn't. Once threshold is reached, voltage-gated sodium channels open rapidly, allowing positively charged sodium ions to rush into the cell. The inside of the cell becomes more positive, shifting the membrane potential from minus 55 millivolts at threshold to plus 30 millivolts at the peak of the nerve impulse. This is depolarization. Shortly after opening, sodium channels inactivate. Since the voltage-gated potassium channels open more slowly, their opening happens at about the same time that the voltage-gated sodium channels are closing. Potassium ions, which are more concentrated inside of the cell, flow out, making the inside of the cell more negative again. Membrane potential drops from plus 30 millivolts to minus 70 millivolts. This is repolarization. Because the potassium channels close slowly, the membrane becomes briefly more negative than resting membrane potential, about minus 90 millivolts. This is hyperpolarization. As the voltage-gated potassium channels finally close, the membrane returns to its resting membrane potential of minus 70 millivolts. The refractory period is right after the nerve impulse begins. During this time, the neuron cannot immediately fire another action potential. The absolute refractory period is when no new action potentials can occur, no matter how strong the stimulus is. This period occurs during sodium channel activation and inactivation. Inactivated sodium channels cannot reopen as they first need to return to resting state. The relative refractory period is when a stronger than usual stimulus can trigger another action potential once sodium channels reset and some voltage-gated potassium channels are still open. Once the action potential starts at the trigger zone or axon hillock, it must travel down the axon to the axon terminals to pass the message on. Unlike graded potentials which die out, action potentials keep their strength as they move along the membrane, a mode of conduction known as propagation. In unmyelinated axons, propagation occurs by continuous conduction, a slower, step-by-step depolarization and repolarization of each adjacent segment of the plasma membrane. In myelinated axons, the signal jumps between gaps in the myelin sheath called nodes of Ranvier. This faster process is called saltatory conduction. This process occurs because voltage-gated channels are distributed unequally in myelinated axons. They are not really found where myelin covers the axon, but the gap between the sheaths have many voltage-gated channels concentrated there, so it's as if the signal jumps as it's being transmitted. Think of it like this. Continuous conduction is like walking step by step, while saltatory conduction is like leaping from stone to stone, much faster and more efficient. As we finish up, let's quickly compare graded potentials and action potentials, since both are essential for how neurons communicate, but they behave differently. Let's first look at where they occur. Graded potentials occur in the dendrites and cell body, where action potentials occurs in the axon starting at the trigger zone or axon hillock. The type of signal that they send. Graded potentials send small, localized changes in the membrane potential, where action potentials are large, rapid reversals of membrane potential. Graded potentials, their strength varies with stimulus strength. They are graded. Action potentials, their strength is always the same size once threshold is reached, that all or none principle. How about propagation? Graded potentials are decremental. They weaken and die out with distance, while action potentials are non-decremental, maintaining their strength along the axon. Graded potentials are used for short distance communication, while action potentials are used for long distance communication. What about refractory period? In graded potentials, this is not present. They can summate to become stronger. In action potentials, this is present. They cannot summate. And then finally, the trigger. For graded potentials, it's the opening of ligand-gated or mechanically gated channels. And for action potentials, it's the opening of voltage-gated channels after a threshold is reached. Graded potentials act as the decision makers. They bring the membrane potential closer to or further away from threshold. Action potentials are the messengers once threshold is reached. They carry the signal the full length of the neuron without losing strength. In our next video, we'll see that the electrical message turns into a chemical one at the synapse and how the body keeps signals from running wild. That's where neurotransmitters come in. If you enjoyed the content of this video, please make sure to subscribe, give this video a like, and turn on notifications so that you never miss out on new content. Thanks for watching, and I'll see you in the next video.